Shri Manjunathaya Namaha. Good morning, each and everyone. We are here in this virtual platform for an IQAC initiative intercollegiate international webinar. Let's begin the event with our institution prayer.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रान्विता या वीणा वरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा पूजिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा न हि ज्ञानेन सदृशं श्रद्धावान् लभते ज्ञानं सत्यमेव जयते असतो मा सद्गमया तमसो मा ज्योतिर्गमया मृत्योन्मा अमृतङ्गमया ॐ शान्ति शान्ति शान्तिः.
Thank you. Now, I express my heartfelt welcome to our delegate speaker, Dr. Melissa, postdoctoral research scholar, Irvine, University of California, USA. I wholeheartedly welcome our principal, Professor Bharati N., IQAC coordinator, Srimati Sukrita KS, advisory board members, Honorable Professor Satyamurthy, research director, STM University, Dharwad, and Professor Ramesh, chief scientist, Mahajanas PD Center, Mysore. I also welcome all the faculties of Life Sciences Department of our college, faculty and participants from various institutions and international participants as well. Now, it's my immense pleasure to introduce our delegate speaker, Dr. Melissa. Dr. Melissa procured her BSc in Biotechnology in 2011 from Manipal University, India, followed by MSc in Medical Biotechnology in 2013 from Manipal University, India. She then completed her joint PhD in 2021 in the field of cell biology, tissue engineering, and photobiology from Manipal Academy of Higher Education, Mahe, Manipal, India, and Queensland University of Technology, Brisbane, Australia. She is a recipient of renowned scholarships like Keystone Symposia Scholarship USA, Higher Degree Research Sponsorship Queensland University of Technology, Brisbane, Australia, and Dr. TMA Pai Structural PhD Scholarship Mahe, Manipal, India. She is the beneficiary of esteemed honors like California Institute for Regenerative Medicine Postdoctoral Training Grant Fellow Award, Interdisciplinary Opportunity Award, NSF Simon Center USA, Travel Award to represent University of California USA in Biotechnology Research Symposium, Best Research Scholar Award Mahe, India, and Best Poster Award at conference held in Gold Coast, Australia. Dr. Melissa has won first prize in poster presentation at Irvine University of California, first prize in DBD ACE KEF Plus 2017 series presentation competition for young scholars, and runner-up in the Department of Biotechnology, India, and National Institute of Advanced Industrial Science and Technology, Japan. Dr. Melissa is the author of more than 7 publications published in peer reviewed journals, participant of more than 20 scientific outreach and engagements. She has been an exceptional and noteworthy presenter of more than 20 poster presentations and talks. Overall, Dr. Melissa is a molecular biologist by training, a passionate cell biologist, a skilled tissue engineer, knowledgeable and highly self motivated postdoctoral researcher with over 10 years of research experience in diverse fields ranging from cancer genomics to stem cells and tissue engineering. With this very brief introduction, I request Dr. Melissa to take over today's session.
Thank you, Dr. Shweta, for the wonderful introduction, and I'll just share my slides. Are you able to see my
Yes.
slides?
Yes, it's clear.
Perfect. So today I'll be presenting on the research developments in stem cell biology. Our bodies are the ultimate factories. Every cell has its specific job to do and is shaped to do that job perfectly. So a muscle cell is only a muscle cell. A nerve cell will only be a nerve cell. A blood cell will only be a blood cell, and a fat cell will only be a fat cell. So the fate of each cell is determined during the embryo stage and then cannot be changed. However, there are these unique type of cells that exist called as stem cells, and these cells have the ability to become any type of cell in the body because they are not specialized. They are characterized by their ability to self-renew and differentiate into various specialized cell types. So self-renewal, yeah. So self-renewal is the ability of stem cells to self-generate, many times contributing to the stock of stem cells in the body. And differentiation is the ability of the stem cell to develop into many different cell types in the body, such as pancreatic cells, muscle cells, nerve cells, blood cells, or lung cells. These then replace the dead and damaged cells in the body. So where do you get stem cells from? Some stem cells are derived from early stage embryo during development, while other stem cells are obtained throughout human life from various sources such as bone marrow, blood vessels, brain, and skin. While we understand more about stem cells, there are certain terms that we should be familiar with. Totipotency, that refers to the ability of a cell to differentiate into all the cell types of embryonic as well as extra-embryonic tissue. These cell types include zygote as well as the early embryonic cells. Next, we move on to pluripotent cells. So pluripotent cells have the ability to differentiate into all the cell types of the embryonic tissue. And these cells include embryonic stem cells as well as induced pluripotent stem cells. Then we move on to multipotent cells, and they have the ability to differentiate into all the cell types of a specific tissue or organ, such as hematopoietic stem cells or mesenchymal stem cells. And unipotent cells have the ability to differentiate into a specific cell type, such as epithelial stem cells or osteoprogenitor cells. To summarize this, self-renewal is the ability of cells to undergo numerous cycles of cell division while maintaining their undifferentiated state. This allows them to replenish themselves over a period of time. What is potency? Potency is stem cells have the ability to differentiate into various specialized cell types. Because they are unspecialized, they can differentiate into specific cell types when given the right type of chemical or mechanical cues. For some stem cells, their fate can be changed and they can differentiate into cell types beyond their original lineage. So this property is known as plasticity. And the behavior of stem cells is often influenced by their microenvironment or niche, which provides signals that regulate the self renewal as well as differentiation of these cells. Hence, they are niche dependent. So, based on origin, stem cells can be classified into three broad types: embryonic stem cells, adult stem cells, and induced pluripotent stem cells. Embryonic stem cells are pluripotent cells that are derived from the inner cell mass of early embryos, typically at the blastocyst stage. They have the unique ability to differentiate into any cell type in the body, making them very valuable for research as well as therapeutic applications. And the major applications of these cells is in developmental study, to study early human development, differentiation process, as well as developmental disorders. And they can also be used for disease modeling to create models of various human diseases, enabling better understanding and testing of potential drugs. And they are also used in regenerative medicine, where they hold promise for repairing and replacing damaged tissues and organs. So there are several advantages and disadvantages of using these embryonic stem cells for research as well as therapy. So one of the advantages is that they can be maintained for long term in culture, and the protocols for maintenance of these cells are well established. These cells can generate most of the cell types, so they can provide an unlimited source of cells. And by studying embryonic stem cells, more can be learned about the early processes of development. But the disadvantages of these cells are the process to generate embryonic stem cell lines is very inefficient, and the therapies using ESCs are new, and much more research as well as quality testing is needed. Undifferentiated embryonic stem cells, which are present in transplants, can cause tumors. And there are strict ethics as well as other regulations, because embryos are destroyed to obtain these cells, and consent is required from female donors. Next, we move on to adult stem cells. These are somatic or tissue specific stem cells, and they are undifferentiated cells found in various tissues of the body. They are important for growth, repair, and maintenance of tissues. So, adult stem cells can be classified into different types based on their origin and differentiation potential. So, hematopoietic stem cells, they are found in the bone marrow, and they can give rise to all cell types of the blood, and they are used in bone marrow transplants. Similarly, neural stem cells, they are found in brain as well as spinal cord, and they can differentiate into neurons, astrocytes, oligodendrocytes, which all contribute to nervous system. And they have potential applications in neurodegenerative diseases and spinal cord injuries. Mesenchymal stem cells, on the other hand, they are found in various tissues of the body and can differentiate into many different cell types such as osteoblasts, chondrocytes, adipocytes. And they have been used in orthopedics, wound healing, and tissue regeneration applications. Epithelial stem cells are found in epithelial tissues such as skin, gut, and airway lining, and are responsible for the regeneration of epithelial tissues used in skin grafts as well as treatments for gut and respiratory tracts. Cardiac stem cells are found in the heart and involved in the repair and regeneration of cardiac tissue. And liver stem cells, found in the liver, as stated in their name, can differentiate into hepatocytes and other parenchymal cell types that are present in the liver. And they are being currently studied for liver regeneration as well as treatments of various liver diseases. Adipose derived stem cells are found in adipose tissues or fat cells and can differentiate into osteocytes as well as chondrocytes. They are being explored for cosmetic as well as reconstructive surgery. Dental pulp stem cells are found in the dental pulp of teeth and can differentiate into dental as well as mesenchymal cell types, and are explored for dental regeneration as well as repair. So these adult stem cells have applications, as I mentioned earlier, they have applications in hematopoietic stem cell transplantation, regenerative medicine, wound healing, neuroregeneration, dental applications, cosmetic and reconstructive surgery, gene therapy, cancer treatment, tissue engineering, drug development and testing, and so on. There are several advantages and disadvantages associated with adult stem cells. The trans differentiation and reprogramming of this cell is possible, but it's not well studied, and they are less likely to be rejected if they are used in transplants. Success has already been demonstrated in various clinical applications, and there are no major ethical concerns. While the disadvantages of using these cells include limitations on the ability to differentiate are still uncertain, because currently it's been thought that these cells are multi or unipotent. They cannot be grown for long periods of time in the culture, and very small number of these cell types are found in each of the tissues, which makes it very difficult to find them and purify them for research as well as therapeutic applications. And there's also lack of technology that is available to generate large quantities of stem cells in culture. Finally, induced pluripotent stem cells are produced through the reprogramming of adult cells, and they can differentiate into any cell type originating from the three major germ layers, including endoderm, ectoderm, and mesoderm. Since iPSCs are derived from adult cells, they do not involve the destruction of embryos, and their use is less controversial compared to embryonic stem cells. They can be generated from the patient's own cells, reducing the risk of rejection and more towards personalized therapy. They have applications in disease modeling and gene editing, where they provide tools for creating models of diseases using cells derived from the patient itself with specific genetic conditions, allowing for better understanding of the disease mechanisms as well as drug testing, and in regenerative medicine applications for generating tissues and organs for transplantation where there is donor shortage. In 2012, the Nobel Prize in Physiology and Medicine was awarded to Dr. John B. Gurdon and Dr. Shinya Yamanaka for their discovery that mature differentiated cells can be reprogrammed into a pluripotent stem cell state. So, how do you isolate iPSCs in the lab? Creating iPSCs involves reprogramming of somatic cells such as skin cells, blood cells, or fat cells into a pluripotent stage using Yamanaka factors. So these factors are really important to reprogram these somatic cells to form iPSCs. These iPSCs, this is how it looks. They form compact colonies, and and they are really small and grow in colonies. They can be then differentiated into any cell type originating from the three main germ layers. So how do you characterize iPSCs? The first step is morphological analysis. iPSCs typically exhibit characteristic colony morphology, with colonies appearing round and compact. Next, we looked at different pluripotency markers and gene expression analysis. So we detect surface as well as intracellular markers associated with pluripotency, such as SOX2, OCT4, TRA160, and so on, NANOG, and so on, using immunostaining as well as flow cytometry, real-time PCR, and RNA sequencing. Then the differentiation potential of these iPSCs is assessed both in vitro as well as in vivo. For in vitro differentiation potential, these iPSCs are differentiated into cells of the three major germ layers: endoderm cells such as lung cells, thyroid cells; mesoderm cells such as cardiac muscle cells, skeletal cells, red blood cells; and ectoderm cells such as skin cells and neurons. For in vivo differentiation, these iPSCs are injected into immunocompromised mice, and tumor formation is observed. This tumor is then excised and then sectioned and stained to see the cell types present, from all the three germ layers present in these sections. Karyotype analysis is also performed to assess the chromosomal stability and to detect if there are any abnormalities that arise during the reprogramming process. Epigenetic profiling is also done to check the DNA methylation to examine if the methylation patterns of the key pluripotency genes have been altered, or histone modification analysis to assess histone modifications associated with active and repressed state of pluripotency genes. So directed differentiation of iPSCs. So this method involves using specific growth factors, cytokines, or small molecules to guide iPSCs towards a particular lineage, often resulting in high efficiency and purity of the desired cell type. There are various factors that affect differentiation. The first one is culture conditions. As you may already know, for culturing different types of cells, you use different media conditions. So similarly, in this case, for culturing different cell types and to promote differentiation towards the desired lineage, there are different culture conditions that you may choose. And then next is growth factors and signaling pathways. So we can add specific growth factors that activate specific signaling pathways that are essential for the differentiation process. For example, for neural differentiation, you add retinoic acid, noggin. For cardiomyocytes, you would add activin A, BMP4. For hepatocyte differentiation, you would add HGF, which is hepatocyte growth factor, basic fibroblast growth factor. And for endothelial cells, you would add VEGF. So the timing and concentration of all these different factors is very crucial during the differentiation process. Finally, the environmental cues. So these were the chemical cues, and these are environmental cues. So using extracellular matrix components such as different hydrogels or bioengineered scaffolds that provide physical and biochemical signals to support cell attachment, growth, and differentiation. So how do you assess directed differentiation? First thing is morphological changes. You'd see the characteristics of the differentiated cell type under a microscope, and each cell type has a specific characteristic. So you would try to match that. And the second thing is marker expression. So different lineage specific markers are expressed at different stages of differentiation, and you check for those using immunostaining, real-time PCR, flow cytometry, and confirm successful differentiation. The final assay is functional assay to test the functionality of differentiated cells. For example, if you generate hepatocyte cells, you would check for either secretions such as albumin secretion or ApoB secretion, or you would look for CYP3A4 activity. For cardiac cells, you would look at patch clamp assay and things like that. There are various challenges associated with directed differentiation of iPSCs. The first one is heterogeneity. Differentiated populations, they are usually heterogeneous, and you would have to do purification to isolate specific cell types. And also the efficiency of differentiation is very challenging and may require a lot of optimization. Finally, long-term stability. So ensuring that differentiated cells maintain their identity and function over a period of time is very important for therapeutic applications. We don't want them to change their morphology or state during long-term cultures. And there are various advantages using iPSCs. They are abundant somatic cells of donor, and the issues with histocompatibility can be avoided. They are very useful for drug development and developmental studies. And the information learned from the reprogramming process, it is transferable for in vivo therapies to reprogram damaged or diseased cells. The disadvantages of iPSCs include reproducibility, maintenance, and efficiency of reprogramming. They can also cause tumors once injected into, similar to teratoma formation that I explained earlier. And iPSCs may also retain epigenetic memory from their somatic origins, which could affect their differentiation process as well as function. And the process of generating iPSCs can be technically challenging as well as time consuming. And there are several regulatory and ethical challenges associated with iPSCs. So, what are the major applications of stem cells? Over the past several decades, major stem cell applications have emerged in research tools. So, they have been used in labs to model diseases, to study development. They've been used in cell therapy, drug target validation. They've been used as unlimited sources of cells for materials for 3D printing, 3D weaving. They've been used as in the drug delivery system as well as toxicology screening. So this is an example of how iPSCs are used to model Parkinson's disease. Here, iPSCs were derived from young onset Parkinson's disease patients, and then they were differentiated into midbrain dopaminergic neural cultures. These neural cultures, they expressed neuron markers such as tyrosine hydroxylase, which is shown as TH over here. And it was observed that the cultures, neuronal cultures, which were obtained from these Parkinson's disease patients, they had higher amounts of this particular protein called alpha sync. So they were trying to find what is the reason for this, and the reduced amounts of lysosomal proteins, it showed that maybe it's not getting degraded, and that could be the reason for increased accumulation of this protein. And they tested degradation rates of and the global transcription function was inhibited for 48 hours using cyclohexamide treatment, and protein was quantified over a period of time up to 48 hours. And it was seen that it was degraded. This protein was degraded in the control while it was still maintained in the Parkinson's disease patients. So this helped the researchers identify important molecular signatures in iPSC-based model of Parkinson's disease, and they were able to identify various biomarkers such as accumulation of this particular protein, dysregulation of various lysosomal biogenesis and function, increases in PKC alpha, which was highly a predictive phenotype, and so it was used as a new diagnostic tool for clinicians. Similarly, several 2D differentiations of iPSCs has led to better understanding of the causes of various diseases, biomarkers, as well as therapeutic targets. But that was an example of monolayer iPSC models. But the cells in your body, they are not in two-dimensional, they are in three-dimensional. So, with the convergence of multiple disciplines, including biology, material science, chemistry, and engineering, there have been advancements in tissue engineering. Adult-derived stem cells, they provide an unlimited source of cells and can be reprogrammed or genetically modified and used to build functional tissues and organs at clinically relevant scales using self-assembly in bioreactors, 3D printing, or decellularized organs, and to construct physiologically relevant in vitro tissue, which is very similar to tissue microenvironment using hydrogels, mechanical substrates, or chemical cues, to model human development, to model disease states, to test various drugs and toxicants. So, with the advancements in tissue engineering, we are moving further away from animal models, which are physiologically very different from humans, and trying to improve the model systems in the lab for all these applications like organ on a chip, or use of robotics, use of engineered materials for clinical applications, as well as various applications in cryopreservation, developing engineered leather as well as meat products. So this is an example of the currently used model systems in the liver. Liver is a very complex organ, and it has multiple cell types such as hepatocytes, cholangiocytes, endothelial cells, stellate cells, and Kupffer cells. All these cells continuously interact with one another and which either occurs because of cell-to-cell contact or by the secretion of various factors that reach cells that are located very far away. While most of the liver development, disease modeling, and preclinical studies have been previously evaluated in cell lines such as HepG2 cells or primary cells, primary hepatocytes, um, people have also used explanted tissues of the liver, but they cannot be kept for long periods of time in culture. People have also used animal models, but they do not completely mimic the human liver, so you cannot really use animal models. So, over the years, many different types of bioengineered liver models have been developed. Uh, so as to have liver cells on hydrogel scaffolds, 3D printing, organoids, as well as microfluidic devices. So this is an example of liver organoids. Hepatic organoids are generated, which comprise of various different liver cell types. They have structures similar to the human liver, and these bipotent hepatoblasts, they give rise to both hepatocytes as well as cholangiocytes. So you can see in green here, albumin positive cells are hepatocytes, and the cytokeratin 7 positive cells here are cholangiocytes. And these organoids are also functionally, they are very functional in the sense that they produce albumin, ApoB, and also CYP3A4, which is very important for the metabolism of drugs in the liver. So it's also been shown that these bile canaliculi network, it is very important for it's a essential feature of the liver that plays a major role in both detoxification as well as transportation of bile to the digestive system. This bile duct-like structures in the organoids, they were able to uptake CDF dye, and using these model systems, um, upon treatment with cholestasis-inducing drug troglitazone, it resulted in, so you can see here, there is very good uptake in the bile ducts, whereas here the bile uptake is disrupted. And also in organoids which were incubated with fatty acids, it's shown that there are signatures of non-alcoholic steatohepatitis signatures. So these model systems have been used to model various diseases as well as drug responses. So Dr. Emmanuel Charpentier and Dr. Jennifer Dodana were awarded the Nobel Prize in Chemistry in 2020 for the discovery of one of gene technology's sharpest tools, which is the CRISPR-Cas9 genetic scissors. Researchers can use these to change the DNA of animals, plants, and microorganisms with extremely high precision. And this technology has revolutionized the molecular life sciences, brought new opportunities for plant breeding, is contributing to innovative cancer therapies, and may make the dream of curing inherited diseases come true. So CRISPR-Cas has two main components. One is the guide RNA, and the other is the Cas9 enzyme. So how does CRISPR-Cas work? So the guide RNA is a short RNA sequence that is complementary to the target DNA sequence you want to edit. So this guide RNA, it directs the Cas9 protein to the precise location in the genome. Once the guide RNA binds to the target DNA, the Cas9 protein, it makes a double-stranded break in the DNA at that location. So this is a double-strand break in that location. And then the cell's natural DNA repair mechanisms are activated to fix that break, such as non-homologous end joining. So this repair mechanism can introduce small insertions or deletions, often resulting in gene knockout. And homology-directed repair, if a repair template is provided, the cell can use it to incorporate specific changes into the genome. So using this CRISPR technology, hepatic steatosis was modeled. So hepatic steatosis is a condition where the liver accumulates excessive fat, causing fatty liver disease, which is mainly due to unhealthy food that we consume. Hepatic steatosis models were generated by feeding these hepatic organoids fats for 5 to 7 days, and with precise gene editing, the effects of gene mutations, particularly PNPLA gene, on steatosis risk can be addressed with high phenotypic resolution. So the yellow color you see here is the fat deposits in the organoids. Treatment with steatosis reducing drugs, it showed that there was reduced drug response with PNPLA variants. And these features allowed the development of personalized organoid platforms paired with drug CRISPR toolkit for NAFLD target identification and testing. And these organoid models facilitate the study of steatosis etiology as well as drug targeting. Another model system I would like to talk about is the blood vessel organoid. So blood vessels are essential in all the organs of human body to supply oxygen and nutrients. And using stem cells, blood vessel organoids can be generated, which were positive for endothelial. So blood vessels are mainly formed of endothelial cells, and these cells are positive for CD31, which is endothelial marker. And it was found that these organoids contained endothelial cells and pericytes, which are the supporting cells which kind of stabilize the vascular networks. So they've self-assembled into capillary networks that are enveloped with basement membrane, just like our human blood vessels. These organoids were then implanted into the kidney capsule of mouse to generate more mature and perfusible artery, vein, and capillary networks. Perfusible means that you can pass dyes through these blood vessels, such as dextrin shown here. So you can pass these dyes through the blood vessels without being leaky, just like how blood flows through our blood vessels. So exposure of these blood vessel organoids to hyperglycemia and inflammatory cytokines such as TNF alpha, IL-6, it resulted in the thickening of these blood vessels and increased collagen 4, which is seen in green here, increased collagen 4 deposition. So in case of diabetic vasculopathy, it's the damage of the blood vessel caused by the complications of associated with diabetes. And by using inhibitor gamma secretase, which is important for the expansion of these basement membranes, we saw there was a decrease. So using this DAPT, we saw a decrease in the collagen deposition. And it was also seen that in diabetic condition, there is leakiness. So when you try to pass a dye through the blood vessels, there is leakiness and it's leaked out outside the blood vessels. So that was rescued in the presence of this treatment. So this is an example of how we can we can study various diseases, blood vessel related diseases, and identify the signaling mechanisms that are responsible for the symptoms of these diseases. Another example is the tear gland. So lacrimal gland is crucial for tear production and ocular health. And lacrimal gland organoids, which mimic the structure and function of lacrimal glands, were generated from iPSCs. And these organoids were characterized by staining for gland markers such as AQP5, as shown here in green, and single cell RNA sequencing. It provided information of what are the different cell types that are present in these organoids. So the next step was to check the in vivo functionality of these organoids. And so they were transplanted and into the connective tissue near the eyes of rats after partial or complete removal of lacrimal glands. The secretion of lacrimal glands, such as human lactoferrin, was found to be increased only in the transplanted organoids. So it was found that transplantation caused improved lacrimal gland maturation as well, which was shown by the polarized nature of these gland cells. So these organoids are very important to study the formation of lacrimal glands and various lacrimal gland related diseases, and for use in regenerative therapies. So another example is gastroloids. So gastroloids are stem cell derived 3D models that mimic early embryonic development in humans. In this study, self-organizing human gastroloids that replicate the key aspects of early embryonic development, particularly the trunk regions, were generated. And it was seen that gastroloids could mimic the complex developmental processes by simultaneously generating both central and peripheral neurons along with the trunk mesoendoderm. So the trunk mesoendoderm is very important for influencing neuronal differentiation and patterning. And this shows us that multiple tissue types interact with one another for proper development. So this studies, by inhibiting this pathway, it was seen that the simultaneous formation of these structures were inhibited. So this shows us that multiple cell types are multiple germ layers are very important during early development, and it emphasizes the utility of gastroloids in studying early human embryogenesis, developmental disorders, and potential therapeutic applications. This study shows the development of mini intestinal organoids that mimic structural functional characteristics of native intestines. So they've used a scaffold guided approach where they have seeded cells and differentiated them into intestinal epithelial cells, and they have seen the accumulation of these LGR positive, LGR5 positive cells at the crypts of the intestine, just similar to the in vivo crypts. And different cell types were found to be formed at different regions of these intestinal mini guts. And these models were then used to study wound healing after laser injury, as well as it was found that parasite C. parvum could undergo their whole life cycle, which was supported in this system, and new oocytes were formed. So this study also helps us understand the host microbe interactions in model systems. Moving on, we go to placenta. Placenta is the lifeline between the mother and the child, providing nutrients and oxygen that is necessary for early development. Previously, it was thought that iPSCs are not able to generate extra embryonic lineages. But recently, it has been shown that iPSCs can be differentiated into trophoblast cells that form the placenta through resetting them into a naive like state. So, trophoblast organoids, which were generated, they expressed most of the trophoblast markers as shown here. And they also secreted various hormones such as HCG, which shows a positive pregnancy test. So, this is similar to the human body. Using these organoids, the authors also studied infections and how maternal fetal interactions are important, and how viruses or infection is transmitted between the two. So these models, since we cannot study these things in pregnant women, these models are very important in providing insights into what happens during early development as well as early life exposures. So similarly, placenta on a chip was also formed using microfluidic devices. The interactions between the invasive cell types in the placenta, called the extravillous trophoblast or EVT, and blood vessel cells were investigated. So these EVTs, they are known to invade and replace the cells in the mother's blood vessels during pregnancy, and the migration of these cells towards the blood vessels, which are over here, can be seen in these images. You can see how these cells are migrating from one channel to the other. So the authors wanted to see, does this migration happen because of the cellular interaction or not? So that's why they have these different culture conditions. So they cultured these EVTs in the presence as well as absence of endothelial cells, and they tried to study the invasion. To confirm that these cells are migrating only in the presence of blood vessels, this study was done, and they can see that there's no migration in the absence of blood vessels. Further, they also used a modified device with increasing distance between the EVT cells as well as vessel cells. And they can see that as the distance increases, the migration decreases, which shows us that there are several factors that are produced or secreted by these endothelial cells that kind of attract the EVTs towards them. And as the distance increases, there is a gradient in diffusible factors because of which they cannot migrate. So what happens is this migration of EVTs is a very important process in early development. And if there are any issues in invasion, it causes complications like conditions like preeclampsia and fetal growth restriction. So, moving on to microfluidic devices. So, this is the picture of a general microfluidic device that you might have seen before. It's a simple three-channel device where you can seed cells at the center and have media on either side. But these devices can also be scaled up. So you can scale up the microfluidic device to have multiple devices in a single plate, seed blood vessel cells like endothelial cells, pericytes, smooth muscle cells, and generate blood vessels on a chip. So this is how the blood vessel networks on a chip look like. And they also pass beads through these blood vessels. So on the side channels, they've added FITC dextrin here in green, and they see how it passes through these blood vessel networks. Here they've added rhodamine, dextrin, and they see how this red color passes through from this side to the other side. It passes through the vascular networks. So using these, so this passing of dyes shows us that these systems are perfusible. And using these perfusible systems, as an example, they've tested anti-cancer drugs on colorectal cancer cells, which have been incorporated within these blood vessels. And they show that 72 hours after drug treatment, how the morphology of some of these blood vessels have changed. So similarly, you can incorporate these blood vessel systems to any cell types or organoids, study the effects of different drugs, or you can model various diseases like arteriosclerosis by feeding them a lot of fat, or test environmental toxicants like nicotine, microplastics, and so on. So the possibilities with these microfluidic platforms containing blood vessels is endless. So, so far we have only seen the chemical cues and how they affect differentiation, and what kind of models do we get by providing chemical cues. So mechanotransduction, it refers to the process by which the cells sense and respond to mechanical stimuli in their environment. So this process is crucial for various cellular functions, including growth, differentiation, and migration. One of the key aspects of mechanotransduction is the role of stiffness, which describes the rigidity or elasticity of the extracellular matrix and surrounding tissues. So different organs in the human body have different stiffness levels. But when, so the brain tissue as well as adipose tissue, it grows in very soft substrates, while the bone is really stiff. But what happens is when we grow these cells in the lab, we grow all of them on plastic, which is very stiff, similar to the bone. Previous studies using stem cells have shown that mechanical environment of the ECM, such as stiffness, is very critical for stem cell behavior. Different tissues, they exhibit varying stiffness levels, and the stem cells can sense this through mechanosensitive proteins and integrins. And studies have shown that if you have a stem cell, you put it in a soft substrate, it is more prone to become neuronal cell. If you put it in a intermediate substrate, it could become something like a muscle cell. Or if you put it in a rigid environment, it becomes a bone cell. So this shows us that the mechanical environment is able to prime stem cells to form certain cell types or towards certain lineages. Using the substrate stiffness as a background and how it primes different cells to differentiate, it will help us understand this study better. So intestinal organoids have previously been generated from iPSCs, but in case of organoids, it's very difficult to like control the structural organization of these organoids as well as cellular compositions. So using various materials, what they have done is they have seeded, they have incorporated intestinal organoid, and using light, they have softened this material in certain patterns, and they have seen that there are cells that migrate towards these soft regions, and this pattern is formed, which can be reproducibly formed, and there are LGR5 positive cells present in the crypts, similar to intestinal tissues. So specific geometric configurations, they can lead to predictable and reproducible patterns in organoid development. So this shows us that the mechanical cues are also very important. So these mechanistic insights were then exploited to engineer microscopic organoids with in vivo tissue architecture using microfabricated hydrogel substrates, and cells were seeded in different geometries to mimic the intestinal structure. By optimizing the tissue geometry, potential applications in improving organoid design for various biomedical applications, including study of cellular processes within the intestine, such as cell shedding and apoptosis, can be studied, as shown in this image. So another example is 3D bioprinting, where perfusible heart tissues are generated. One of the major challenges in tissue engineering is that you cannot generate organs that are as big as human organs. This is because there is a lack of diffusibility at the core, because there's no oxygen and nutrients at the core, and the cells start to die off, as you can see here. So the cells in red are the dead cells, while green represents the live cells. So tissue engineers have developed perfusible systems that provide these nutrients and oxygen to the core of the tissue, and increase cell survival. So people have developed innovative printing method known as Swift, which was developed for creating organ-like tissues that integrate various networks. So here you can see how the printing is done in a granular matrix. So the first example was without ink, and this is in the presence of sacrificial ink. You can see how it moves through the granular matrix. Next, I would like to show the printing process, where you can see how blood vessels are printed within these structures using Swift printing method. And these blood vessels are found to be perfusible, and there is very less cell death at the core of these tissues. So this way you can make big tissues without having cell death at the center. These techniques were also used to generate perfusible cardiac tissue. As you can see here, there is cardiac contractibility, and it was maintained in these structures. So this study marks a significant advancement in tissue engineering highlighting the potential for further optimization and in vivo integration of these complex tissues. Next is a kidney tissue. So the study demonstrated the successful bioprinting of 3D convoluted renal proximal tubules integrated into perfusible microfluidic devices, which closely mimic the structure of the natural kidney tissue. The engineered tubules, they expressed various proximal tubule markers, and people also saw polarization of these cells. Perfusion of these tubules enhanced cell viability and functionality over time, and it provided a robust platform for drug testing and toxicological assessments. Another example of tissue engineering is the human hair follicles using a biomimetic developmental approach, which is a promising area of research to develop effective treatments for hair loss. So these 3D bioprinted molds, as you can see here, were seeded with different cell types in the hair follicles, and it was seen that addition of keratinocytes to these systems led to the integration of these constructs with the surrounding tissue and generation of hair follicle, which you can see over here with the hair shaft formation. Further, to reduce the cell death, these systems were vascularized and then transplanted into nude mice, which usually do not have hair. As you can see here, with this technology, they were able to generate and grow hair in nude mice. This is an example of technologies and advancing regenerative therapies in hair loss treatments and enhancing our understanding of follicular biology. So far we have looked at the different applications of stem cells in research, disease modeling, and potential therapy. So the most commonly used stem cells in therapy are hematopoietic stem cells, and it is very common, and it's commonly known as bone marrow transplant. It involves administering healthy hematopoietic stem cells to patients with either dysfunctional or depleted bone marrow to augment bone marrow function. So these hematopoietic stem cells can generate various different cell types that form the blood. They have clinical applications in multiple myeloma, in various leukemias, in sickle cell disease, thalassemia, anemia, and so on. Mesenchymal stem cells are also being used in therapy, and it's an emerging area of regenerative medicine. And because mesenchymal stem cells can be generated from numerous sources, such as all these, and they have the ability to treat various conditions because they can differentiate into multiple cell types and because of their immunomodulatory properties. So they have been used in applications such as osteoarthritis, bone fracture, spinal cord injury, chronic wounds and burns, rheumatoid arthritis, and so on. iPSCs have also been used in therapy, and it's an innovative approach in regenerative medicine that uses unique properties of iPSCs to develop personalized treatments for various diseases. So a biopsy of skin can be taken from a person. These adult somatic cells can be reprogrammed using the Yamanaka factors into iPSCs, and then iPSCs can be differentiated into many different cell types and could be injected back into the same patient. So this allows for patient-specific therapy. And it has been used in ocular diseases, gastrointestinal diseases, diabetes, liver failure, spinal cord injury, and so on. So this shows us the market for iPSC-based patent publications is increasing rapidly, but the clinical trials over the years are not increasing at the same pace. This could be due to the high cost, time, and strict regulations, as well as ethical concerns that require strict monitoring of the cells derived from stem cells due to either tumor formation or the source of cells or the culture methods, characterization of these cells, as well as rigorous animal testing. The ethical issue is another factor that could impede these development, and Different countries have different levels of permission regarding these cells and applications to permit in clinical settings. Stem cell biobanks are also becoming very common these days, where they are repositories that collect, store, and manage stem cells for research, therapeutic, genetic diversity, quality control, and clinical applications. So, when you have stem cells, they are collected from various sources, processed to ensure the viability and purity before storage. They are then typically stored in liquid nitrogen at very low temperatures to maintain their viability for extended periods of time, and regularly they are checked to ensure these cells are viable and uncontaminated. The stored stem cells are often characterized for their pluripotency and differentiation. These biobanks provide stem cells to researchers and clinicians for studies and therapies, often following all the ethical guidelines and regulations. They maintain detailed records of donor information, including demographics, health data, to support research and ethical compliance related to informed consent, privacy and confidentiality, and regulatory compliance. There are different types of stem cell biobanks, such as the public biobanks, which is open to all researchers worldwide, and they have broad range of resources. Private biobanks usually charge fees for access to their stem cell lines, and they focus on specific applications or specific type of cells and provide customized services. There are also disease specific biobanks as well as genetic diversity specific biobanks, which have samples from individuals with specific diseases to facilitate targeted research and therapeutic development, as well as cord blood banks, which collect and store stem cells from umbilical cord blood, which are rich in hematopoietic stem cells and used for treatment of blood disorders. So, continued improvements in regenerative medicine techniques, it could lead to effective treatments for complex diseases, leading to advanced therapies. iPSCs enable patient-specific therapies, which minimizes the risk for rejection as well as enhances treatment effectiveness through personalized medicine. And it paves the way for lab-grown organs for research applications as well as addressing transplant shortages through bioengineering approaches. And better understanding of cancer stem cells could lead to therapies that are specifically targeted towards these cells, reducing the rates of reoccurrence. And using interdisciplinary technologies for clinical application, uh, data analysis, and drug discovery could lead to improved outcomes. So, with this, I would like to thank you for your attention, and I would be happy to take any questions.
Thank you, Dr. Melissa, for an excellent informative session. Anyone having queries can now unmute and introduce your name and designation and ask the question.
Can I have a couple of comments?
Yes, sure.
It's not questions for your eloquent lecture, but compliments for your uh, all the explanation that you gave. In fact, I was wondering, so much of information has been covered within a short time. I feel that whatever you presented today is worth of one course in one semester.
Yeah,
So,
I try to cover as many topics as I could to help the students understand what all applications these stem cells have.
yeah, fine. The thing is that it was so elaborate that you covered almost all the organs in the body and the related stem cells and their applications and how best they are used, and with authentic scientific data. See, you can talk for an hour or more, but with authentic scientific data and also the illustrations that you provided were really, really impressive. Well, I
Thank you so
am
much,
happy.
sir.
I'm happy to see that you mentioned you took the name of John Gordon during
Mhm.
your lecture. In fact, yeah, I was with him for nearly 10 days when he was in Mysore, interacting with him. It was my privilege to be with him at that time, learning many things. Yeah, fantastic. Melissa,
That's
congrats.
amazing.
And it was a,
Thank you.
very resourceful lecture. And I told you that it is uh, good to organize for one course in one semester. Uh, 30 hours of teaching can be done in this. And he also covered the latest information with regard to biobanks and stored umbilical cord. And I think that's a very good source of embryonic stem cells. If you collect the umbilical cord and establish a biobank, I think apart from some ethical issues of uh, permissions from the parents or whoever, it does not involve any kind of ethical committee issues. Yeah, just the
Yes,
permissions
because
are
I
enough.
think because they are adult stem cells, they are more relaxed with the regulations. I think
Correct.
with regard to embryonic stem cells and iPSCs, they are very scared because embryonic stem cells, you get it from embryos and you don't want to destroy life. That's what
Exactly.
we think. And
Correct.
with
Correct.
iPSCs, they can form tumors because you use different factors to reprogram them. And when we try to genetically change a cell type that is naturally occurring, there might be so many side effects that we do not know about in the long term.
Exactly. See, when these cells are injected back, regulation becomes a great problem.
Yes.
Where they have to stop, how they have to stop, etc., etc. And regulating from outside also is a great challenge. So maybe in the years to come, all these things will be resolved.
Yes,
With,
because I think earlier people used to grow these iPSCs on Matrigel and
exactly. Yeah. Yeah.
the ECM like that. So Matrigel usually comes from rats. So
Mhm.
it's you cannot inject something that comes from rat, which when your cell comes in touch with something from a rat, you cannot inject that back into a patient. All that as well. But now people have moved away from those sort of systems. They have more defined systems without
Mhm.
Matrigel and things like that.
Mhm. Mhm. Well, these biobanks are working as usual and commercially.
Yes,
Yeah. I don't think there are such biobanks in India.
I don't think so. I believe
Yeah, I have
there
not
are
heard
no
so far,
biobanks.
excepting that umbilical cord is maintained for later use. That also is
Yes,
not very popular, although
I
it
think not
started
everyone does it because it the and is also
maintenance.
quite low, so not everybody knows about it. And
Mhm.
these could be very helpful because they have hematopoietic stem cells. These umbilical cords are very rich in hematopoietic stem cells, which can treat various blood related disorders and things
Yes,
like that
yes, yes.
later in life as well.
Yeah, although such things started 20 years ago, there was a sharp increase in the people who were interested in that, and then later on it silently died.
Yes,
It did not continue.
I think it was not feasible because of the high costs and maintenance of those facilities over the years.
Mm, mm, mm. Maybe, yeah. Okay. Thank you,
Thank you.
Melissa. It was very wonderful narration of whatever. Yeah. Thank you.
Thank you, sir.
Thank you, sir. Is there any other who has questions or inquiries or any doubts? You can even type in the chat box.
Hello, ma'am.
Hello.
Hello.
Yeah, ma'am, I had a question.
May I know name and designation?
Myself, Akhila. I'm currently a first year MSc Biotechnology student from Mahajanas College,
Okay.
Mysore. Ma'am, I was currently practicing how to write a review paper, and I chose a topic called menstrual stem cells. And
Mhm.
like you said, the umbilical cord is rich with hematopoietic stem cells, and I've been reading few papers for this review work. So I wanted to know, like in the papers, they have said that, rather than extracting stem cells from the bone marrow, which is very painful and risky, the menstrual stem cells have given more promising results compared to the bone marrow stem cells. So can you just talk something about it? Like, I want to know more about it.
I think menstrual stem cells are more recent, so it's not been used that often. I think I am not very familiar with menstrual stem cells actually.
In due course, work will pick up on this on these lines. Many people might be interested in collection of stem cells and
Any more question
how
is
far it can be
concluding?
used in treating whatever is also important. It may not be used at all. I do not
ಎಲ್ಲ
know.
ಹೇಳ್ತೀರಾ ನೀವೇ?
Collection is one, and maintenance, and then purification, and all those things have to be taken care. Maybe in the due course of time, Akila, you can build a project on that and then apply for a postdoc elsewhere.
Yeah, sir, sure.
Hello, ma'am. I'm Shrupta. A very nice
Hi,
talk, Melissa,
Shrita.
ma'am. Hi, ma'am. Very nice one, ma'am. Ma'am, I have one doubt, ma'am. For the cervical
Mhm.
cancer modeling, ma'am, if we want to create a 3D model, which type of model you suggest, ma'am? Is it a matrigel based or any other mold which you showed? Such type of model is good or
Yes, ma'am.
matrigel based one is good, ma'am?
So I think based on your application, so what exactly do you want to study? Do you want to study
Host
the
microbiome
early
interaction.
so host microbial interaction. So if you want to study different factors interaction, I would suggest microfluidic devices are the best because with microfluidic devices you can have multiple cell types
Can I ask
together,
you?
and you can also flow all these factors and then study the
ಇನ್ನು
effects.
ಯಾವುದಾದ್ರೂ ಇದ್ದರೆ ಇದರಲ್ಲಿ
What are
ಮಾಡ್ತಾಯಿದ್ದೀನಿ
the effects on one
ಬಂದ್ವೇದಾದರೆ
cell type, or what are the effects, like combination of
ಕ್ವಶನ್ಸ್
different cell
ಬರ್ಕೊಂಡ್ರೆ
types. So
ಫೋಟೋ ತೆಗೆದ್ಬಿಟ್ಟು
I
ಇಲ್ಲಿ
would suggest microfluidic systems for those.
Okay, yes, ma'am. Yes, ma'am. Thank you very much, ma'am.
Thank you.
There is one more question from Diana. She is a research assistant and pursuing PhD on the topic leprosy. Her first question is what type of stem cells can be isolated from urine? And the second question, how do urine derived stem cells compare to other types of stem cells in terms of differentiation potential?
So I think with regard to urine, um, I'm not sure if there are tissue specific or adult stem cells present in urine. I'm not very familiar with that, but I'm sure that if there are any other cell types in urine, you can reprogram them. So basically, you can reprogram any cell type that is present in your body. So you can reprogram urine cells as well to form iPSCs, and then these iPSCs can be differentiated into any cell types that you can study, I think.
Okay, okay, thank you. Anyone
Mhm.
else?
Reprogramming of, see, terminally differentiated cells as such can undergo the process of de-differentiation and further become the iPSCs for, yeah. Yeah, one of my close friend who is a medical doctor has been successful in reprogramming the skin cells into neurons. So that was
Yes.
a wonderful contribution, and he continues to do so. So like that, many applications are envisaged, which becomes useful for human race or for commercial purposes also. Yeah, that's a good kind of work, and abilities can be enhanced.
Okay, thank you, sir. If there are no more questions, we can end the session. I Okay, there is Akshita P. How promising is stem cell therapy in treating blood disorders?
I think it's very promising because there are a lot of treatments that are already going on with regard to blood disorders like sickle cell anemia, thalassemia, and all of those using hematopoietic stem cells. So it's not only promising, people have already started using them as therapy, and there are a lot of clinical trials as well going on for these stem cell therapies. But it's not very common in all the countries because of the ethical regulations as well as the permissions that are allowed.
Thank you. Okay, now we are at the end of the session. I
There are a couple of more questions, Shweta.
Yeah, yeah. I'm checking.
Well, Akila Rane has got one question,
Okay.
and Banu Prakash.
Yeah, Banu Prakash, her question is, by using hematopoietic stem cells, can we treat other than blood-related diseases?
So hematopoietic stem cells, they are adult stem cells, and they can only form different cell types within the blood lineage. So they can only produce different types of blood cells. They cannot produce any other cell types. But you can
Right.
also differentiate these and reprogram them into iPSCs and then
Mhm.
get many other cell types from these cells.
Sure. Akila has question.
Akila has actually requested Dr. Melissa to share her email ID if she doesn't mind,
Okay.
so that
Oh,
she
sure.
can
I
definitely.
can do that in the chat.
Yeah.
I'll.
Okay, sure. So in future she can ask any questions or help regarding her research work. I think there is Rakshita P. Can stem cells be used to cure genetic disorders like thalassemia?
Yes, so a lot of work has been done in thalassemia as well. People are trying a lot of clinical trials in different countries where they have also genetically modified these stem cells. And so, with CRISPR technology, that has more promising features where you can precisely you can edit different genes which are disrupted in certain conditions, and then you can rectify them and you can treat many diseases with that. So I think animal modeling and all of that has already been done, and people are using it for clinical trials right now.
Thank you. Okay. Seems like there are no more questions. Finally, I express deepest gratitude to Dr. Melissa for acknowledging our request, and I wish good luck uh, to you to contribute more and more in science and in the biotechnology field. Thank you so much.
Thank you so much, ma'am, for giving me this opportunity.
Thank you. I thank Dr. Satyamurthy for referring Dr. Melissa for today's event. Thank you so much, sir. I express my deepest gratitude to our advisory board member, Professor Ramesh Mahajanas PG Center, Mysuru, for his consistent, marvelous support and guidance. Thank you, sir. Thank you so much. Thank you for being with us here in the
My
Beings
pleasure.
Virtual
Thank you.
platform.
Yeah. Thank you.
I thank each and everyone who
Yeah.
participated and supported the event. Thank you. Thank you, one and all.
Thank you.